Hi, Susanna. Thank you for coming. Today we have the microbiome room. What happens after taking antibiotics and what's the best way to address that?
Hi, Katarina.
Hi, Victoria. How are you today?
I'm good, thank you. How are you doing?
I'm good, thank you.
I'm just finishing up at the market. So,
Oh, yeah, go ahead. I'm just sharing the room and setting things up, so we still have a few minutes.
great. I just bought some yogurt because I'm trying to keep with the microbiome theme.
That's perfect, and I love yogurt, so yeah. Hi, Daniel. Hi, Suzanne. Hi, Venkat. Thank you for coming. Our guest speaker will be here in a few minutes, and today's room is about the gut microbiome and especially what happens after taking antibiotics and how we can address, to basically heal our microbiome again. So, our researcher is from MIT. Yeah, he will be here shortly, and thank you everyone for coming.
I just PTR'd the yogurt because I think it's so pretty, the package.
Oh, it is. That's a nice one. I also bought yogurt this week at the farmer's market from a local farm. It's
So,
amazing.
yeah, this is a local one too. That's great.
I also I don't like cow's milk. Like, I like yogurt and everything and cheese. I don't like milk, but when it's truly fresh milk, like when we used to go to our friend's farm in Switzerland to get the real fresh milk, then I loved milk. It's amazing.
Oh my gosh, what a difference! What kind of cows? Because here it's hard. Like I know in Europe, it's often Jersey milk or Guernsey even. Sometimes here it's mostly Holstein milk, but that milk is so much richer. I wonder, do you know what kind of cows they were? What color they were?
They were
They were.
brown, like dark
Yeah,
brown.
so they're probably Guernsey cows. The Holsteins are black and white. Those are really common here.
Yeah, their milk is really like very creamy. It's amazing. And I think it has a lot of microbiome. Like it's very good for you, I think, to drink like not pasteurized milk. It's a completely different taste. It's like it has nothing to do with the pasteurized milk taste, I think.
Oh my gosh, no. Yeah, it's so rich and it has flavor and a scent. It's like a milk experience.
And we used to bring so after a while, I was allowed to help. So to help out with the cows and other farm related stuff. So we used to bring them up to the to the Alm, up high, up on the mountains, and then bring them back. I loved
You
it.
brought the cows? You
Yeah, I
Oh my
loved
gosh.
going there.
Oh my gosh.
Yeah, it was amazing. Like we used to go every fall and spring, but then later on, I don't know if they sold the farm or whatever happened. Then they didn't have a farm anymore. They had like a house and close to a city in Switzerland, so that was still nice. It was not the same.
So we need a field trip.
Oh, definitely. Oh, yeah, I love it. Just the smell, like it's so good.
Oops. Okay, I'm gonna go check out now.
Okay. Hi everyone. We will start in 5 minutes. Thank you for coming, everyone. We will talk about um, Dr. Andrés Ruiz. He's a researcher at MIT and Professor James Collins' lab. And he will talk about his latest paper that he published about the gut microbiome and what happens after taking antibiotics, and how the best way to basically repopulate the microbiome, so we won't have as much risk of inflammation states and having other disorders related to have a poor gut microbiome. So, hi Jamie, how are you?
Hello, everyone. How's everyone today? Katharina and Victoria, you both here? Oh,
I'm
hello.
here. Hello, how are you? Hi,
Hi,
Jamie. Hi,
Andres.
Katerina.
How are you? Long time no speak.
few hours.
How are you? Sorry, I was quiet for a moment. I was getting the link for the presentation now. We had the paper up so people could look at it. So, yeah,
Nice.
welcome. Thank you for coming.
Thank you for having me here. I'm very happy to be here to share our work.
We're very, very happy to have you here, Andreas. And Andreas, is that correct?
Andrés, yes.
Andrés, yes, Andrés. Okay, thank you. I was saying before I was looking at your paper, it's quite exciting. Oops, I just dropped. Sorry, I'm just going to wait for a second.
Hello, Andres.
Hi, Victoria.
Welcome. Nice to meet you.
Nice to meet you. I like your profile picture that has some yogurt in there.
Yes, thank you. I was trying to stick with the theme.
That's good.
I might change in a bit, but for now, I'm a beautiful blackberry yogurt.
Sorry about that. I just had a bit of a spillage
Hi,
there.
Jamie.
But yes, hi, Victoria. Your paper's really, really interesting. The engineering biotics it's quite exciting that the potential for what it is you're doing here in this paper. It's quite exciting.
Yeah, we're very excited to move these forward for the benefit of patients, because we really think that there's a highly unmet need here that we need to cover.
Very, very. I would say it's preemptive, but it's not before time that we have something that can help from this issue with and too many people taking antibiotics and building up immunities to them and things like that.
Correct. To resistance, yeah.
I was almost going to start asking you questions, but I don't want to get
Yeah.
myself.
Let's start the room.
Yeah. Oh, yeah, absolutely. Yes.
Yeah, thank you. So, thank you everyone for coming to Science Society, and of course, a special thanks to you, Andres. And let me give our audience a little bit of information about you first. Dr. Andres is a microbiologist with expertise in synthetic biology and genomics. He received his Bachelor in Science and Master in Science in Microbiology from the Universidad de los Andes in Bogota, Colombia. And he did his PhD in microbiology at MIT, Massachusetts Institute of Technology. And under the membership of Professor Sally Kishon, and for his postdoc, Dr. Andres joined the laboratory of Dr. James Collins, where he is right now at MIT, and he is applying synthetic biology approaches to endow bacteria with diagnostic and therapeutic capabilities. And he's developing an engineered live biotherapeutic platform that targets the gut microbiota for the prevention and treatment of human disease. At the WIS, Andres leads a WIS validation project aiming at using this platform, this engineered live biotherapeutic platform, for the prevention of antibiotic-induced dysbiosis and its associated pathologies. So thank you so much for coming, and Victoria will ask you a more general question first, and then the stage is yours for the presentation. Thank you.
Thank you, Katerina, for the introduction.
Yeah, thank you, Katarina, and welcome, Andres. We are so happy to have you to carry us into the body of your talk. It's really nice to hear a little bit about you. And so if I have a question that can just have a brief answer, if that's okay with you, if you can give us a bit of information about when you first noticed that you felt a particular connection to science, and thinking back into your life, it could be in your childhood or even sometime during your schooling. But if there was a moment that you felt like you noticed that science was something that was really something you carried an affinity for, that's my question.
Yeah, I think it's truly for most scientists that this happens during childhood. And in this case, my connection to science and to biology in particular was when I growing up. I grew up near Bogota in a farm. My family has a farm, so I was surrounded by nature, and I saw like a lot of transformations happening in the fields. Like with plants decomposing, and I didn't know why was that happening, why they were disappearing, and starting to think about like these type of transformations that happen around us that are somehow like magic, that are mediated by microorganisms, and this is when I discovered microbiology, and then I was fascinated how things that we cannot see can actually transform our environment and have an effect on our lives. That was like my moment when I discovered my passion.
My gosh, thank you so much. It's fascinating to hear when people's answers are associated with play, with non-directed activities. And you had that experience, and also how rich nature is, and how much of an education nature can provide for us if we're just set free there. So thank you so much for your answer. And at this point, you're welcome to step in to deliver the work that you're here to share. And at the end, if that's okay with you, then we can have a brief Q&A. So relax and enjoy. And we have the PDF pinned at the top. So Andres, the mic is yours.
Thank you, Victoria. So this is informal, and please feel free to ask questions at any point during this conversation. Just to follow up on what I was saying about the role of microbes in the world and why it fascinates me. For my PhD, I was studying microbes that live in the ocean, and it was very interesting to see how their activity, their metabolic activity in the ocean, basically controls the biogeochemical cycles. Like they are, of course, in charge of producing oxygen, consuming CO2. There is a bunch of nitrogen and phosphorus cycles that happen there, and they have a huge impact in our lives. And after I finished that research, I wanted to turn into us as human beings, and seeing how microbes would affect our functioning, our bodily functions. And this is when I started to read about the microbes that live inside and on us, that are called the microbiota. These tremendous amount of diverse microbes that we harbor, and how important they are for us. And this is when I discovered that actually, these microbes are not regarded only as, living beings that are inside us or on us, that just happen to be there, but actually that we have co-evolved with them. Our bodies have used them over evolutionary times to basically outsource many functions, to the point that we consider now them as another organ, as for example, a liver, or lungs. They are accomplishing functions important, such as any other organ in our system. So to me, it was incredible to see that, given the importance that we have seen in those. And if you go to the slides that I have provided, probably the second slide, our microbiota, the studies that have emerged in the last, I would say, 15 years, first on association studies, and then more mechanistic studies, have revealed that our microbiota, a diverse group of microbes that live inside us, really accomplish important functions. For example, pathogen exclusion. They prevent that they invading pathogenic bacteria would colonize our guts. They are also important for newborns and infants for their immune system maturation and the functioning of the immune system. And as well, they intersect with other systems in our body, not just the digestive, but also like the gut-brain axis and many others. So it is true that these microbes are really important to us. And the problem that I saw here is that that people have realized before as well, is that certain types of lifestyle choices in our modern life can really hurt these microbes that really in there. Specifically, I'm talking about medications and diets, particular type of diets that can reduce the diversity that we have in there and change the balance that we have nurtured throughout our lives in our guts. And this is what we call dysbiosis. Once this group of microbes, their population shift and their metabolic capacities change due to the action of medications or the diet, it becomes dysbiotic. That is an unbalanced state. And in that state, the microbiota is the gut is more susceptible to diseases, for example, opportunistic infections. You have heard about Clostridioides difficile, which is a very common hospital-acquired infection, but it also in the long term predisposes you to higher risk of developing inflammatory or metabolic diseases. And even some dysbiotic states can affect how well you might respond to, for example, some types of cancer treatments. So it's really important to preserve this composition that we nurture throughout our lives from the beginning, that we are born without any microbes in our gut, and we start eating. We collect and we create a very personalized collection of microbes inside us. So seeing the importance of these microbes, I wanted to see if there is a possibility that we could protect this important group of microbes when we take antibiotics. And the reason is that antibiotics cannot differentiate between good and bad bacteria, right? The mechanism of action of antibiotics work the same for a pathogenic bacteria that is causing an infection and for a commensal bacteria that lives in your gut. And the reason is because once you take the antibiotic and the antibiotic goes into circulation in your body, it eventually will reach the gut through biliary excretion. And then the microbes will be exposed to the antibiotic and it's going to induce this dysbiosis state. And as you know as well, it's not only the microbes that you kill that are important, but also the ones that survive, because the ones that survive are the ones that can withstand the action of the antibiotic. So we're selecting for antibiotic resistance every time we take an antibiotic in our guts. And you have heard about these, like antibiotics are life-saving medicines and we still have to use them. And in no way I'm saying that antibiotics are bad for you. They are necessary medicines. But what I'm saying is that we have to do better in making them more safe and safer every time that we take an antibiotic. And the reason is that for most of the infections that a doctor prescribes, there is no need to have the antibiotic action in the gut, right? If you have an ear infection or if you have pneumonia, or if you have a urinary tract infection, there is no need for the gut microbes to die because of the action of the antibiotic. So that's why we created a strategy using engineered live biotherapeutics to protect the gut. So that's our slide number 4 there, if you want to have a sense of what might be happening. So doctors in the scientific community realize that this is a problem, but they haven't found a real effective way of preventing this damage to the gut microbes. Usually probiotics and fermented bacteria that are isolated from foods are prescribed with the antibiotics or after the antibiotics, but the reality is that they have no capacity of protecting the native microbes. And in fact, there are some studies that say that they might delay the recovery of the microbiota after the antibiotic treatment, the delay of the native microbes, the indigenous populations that we need in there. And but overall, they have shown like moderate efficacy and empirically there is some signals that say that they are safe and a good platform to use, to improve what we can do to protect the gut microbiota. And this is exactly what we did. We used synthetic biology approaches to endow these bacteria, these probiotic bacteria, with the capacity of protecting the other microbes in the gut from the action of the antibiotic. So the idea is that whenever you need to take an antibiotic, at the same time, you can take a pill that contains these engineered live biotherapeutics. This is how we call them. So they would populate the gut and they would perform uh, localized degradation of the antibiotic in your gut, protecting the good microbes that live inside of your intestinal tract while the antibiotic circulates in your body, um, clearing the infection that you need, for example, for an ear infection, for the pneumonia, even prophylactically if you need to take them. Of course, if the infection that is happening in the first place happens in the gut, you wouldn't use this protective intervention because you need to clear out the gut from an infecting pathogen. So this application is for every time that we would need to treat a patient that doesn't have any gut infection. So at the end, for those instances, you can end with an intact gut microbiota. So as you know, there are different classes and different mechanism of actions of antibiotics, and everyone act a little differently. So we wanted to focus first on one group of antibiotics to start with, and we decided to go after beta-lactams, or most people know them as penicillins. It's a structural class of antibiotics that are the most relevant in the clinic. They make up to 65% of the antibiotic used in the United States, and they amount to more than 150 million of prescriptions in the US. And within these groups, we have the penicillins, cephalosporins, and carbapenems, which are like incredibly life-saving medicines. So we have to make them safer for the patients and for everyone. So the key challenge here is that if you're going to make a probiotic, a bacterium, express an enzyme that can degrade an antibiotic, you have to do it in a safe way, because normally those enzymes are present in bacteria that are resistant to the antibiotic. This is where we found them. They have evolved a way of surviving these antibiotics. And usually these are pathogenic bacteria, or most notably they are pathogenic bacteria, although they can be present in commensals as well. And the way they work is that this enzyme called β-lactamase that is capable of breaking down these penicillins, it sits in the cell wall of the bacteria, and it degrades the antibiotic as it comes to the cell to bind to its target. So this confers antibiotic resistance, and because of this great selective advantage that they confer, they are easily spread through horizontal gene transfer, right? So that's why we're seeing more and more reports that like a new pathogen acquire a resistance mechanism that now makes it resistant to last resort antibiotics like carbapenem. So it is a huge problem. And of course, if we're going to put it in a probiotic bacterium, we want to make sure that we're not going to keep spreading this capacity of degrading antibiotics to other microbes that we don't want them to have. So that's why we utilize this synthetic biology approach, which is basically using engineering principles to take pieces of genes of DNA and assemble them in a way that would confer a new function that works in a particular way in a bacterial cell. So in this way, we created an engineered live biotherapeutic that, instead of degrading the enzymes in their cell wall, they do it in the extracellular environment. Basically, they secrete the enzyme to the outside environment, so it will be for the benefit of the whole microbial community. It's basically a public good, and at the same time, it doesn't confer beta-lactam resistance to the producer microorganism. It is still sensitive to the antibiotic, and the survival is depending on the cell density. And at the same time, we came up with an idea on how we could encode these genes in a way that it would not be transferred to other bacteria in the microbiota, and specifically to pathogens. And these biosynthetic refactoring of the beta-lactamase is what makes it safe, and makes us confident that we're not going to be spreading this trait to other bacteria. And basically the reason is that it would be invisible to natural selection. This is a trait that can only be selected under specific laboratory conditions, and under the natural environment, there is not enough selective pressure for it to be advantageous in another cell. So this is the gist of the idea of how we can make it contained within our particular group of organisms and not transferable to anyone. So with these set of safeguards in place, we wanted to actually see if by producing this enzyme through, let's call it probiotic strain in the gut, we would be able to get the effect that we want. And the effect is that we could treat a patient, and after the treatment, the patient would have an intact gut microbiota, despite the fact that the antibiotic was circulating in the rest of the body. So we used a mouse model for that, and this is what we published in recently, a couple of months ago, in the journal Nature Biomedical Engineering, where we did a proof of concept in a preclinical model of mouse, where we showed that actually our engineered bacteria has antibiotic degrading capacity in the mouse gut, that you can see the degradation of the antibiotic specifically in the gut, and it does not change the concentration of the antibiotic that is circulating in the blood, which is absolutely necessary to maintain the efficacy of the treatment of the antibiotic that you are prescribing to a patient. And then through DNA sequencing, we showed that it can preserve the diversity of this microbiota. It maintains the initial structure. And importantly, as I was mentioning before, it prevents the enrichment of new antimicrobial resistance genes in the gut, which is very important, to curb the emergence of new variants that could be resistant to these antibiotics. So that at the DNA level we could detect, and then we wanted to see more on a clinical readout, how you could see these how these would be useful for in a clinical setting. So we decided to test if this intervention could maintain one of these functions of the microbiota, that is the colonization resistance. So what we did is we treated the antibiotic with the mouse with the antibiotic, and at the same time we provided this protective intervention, and then we challenged them with the pathogen C. difficile. And we saw that the ones that received the protective intervention had a microbiota that was healthy and diverse enough that could keep excluding the pathogen from the gut, and we didn't see any infection, while the ones that received a normal control Lactococcus lactis, which is the probiotic that we have been using, did have a complete infection there. So this speaks as well that the normal wild type probiotics that we use, like, do not actually protect against a possible C. difficile infection. So it was very nice to see that we had this 100% response in this preclinical model, and now we're looking to translate these findings for humans by conducting more experiments and then going to human clinical trials for that. So this is what we have done. And before I stop, so we can have more of a conversation, I just want to say, like, if you're looking at the slides, the last slide that I have in there is that we have to start thinking about the microbiota and protect it like we protect other major organs. If you go to the sun, on a weekend to the beach, like, you're gonna put on sunscreen to your skin, right? Because you know that it can harm you even though the effects you're not going to see immediately. The same, we know that whenever we go to have some x-rays, we're going to cover the rest of the body where the x-ray is not needed. So we prevented the damage of the ionizing radiation that the machine can give you, right? And in the same time, in the same fashion, we should be thinking about protecting our gut microbes from the action of the antibiotic. They are so important, and so many consequences can arise from these dysbiotic states that it is truly important that we make them safer for every patient that needs them. So I'm going to stop there, and happy to answer any questions or comments that people might have in the room.
Thank you so much for this great presentation and for sharing this very important research with us. Thank you for also spreading the awareness how important it is to protect our microbiome. And this is amazing work and an amazing talk. So I saw that Eli, you had a question. Go ahead.
No, actually, no question. I came a little too late to ask intelligent questions.
So maybe just a question for the general, for everyone here that might be interesting. So you mentioned that maybe, usually people give, or doctors now give when they prescribe antibiotics also, probiotics, prebiotics. But you mentioned that it's not really known if that's a good idea, because that doesn't reflect the variety we used to have. So do you recommend not doing that acutely, or until we have a better option doing that, or what would you say?
I'm not a medical doctor, so I cannot give like any formal recommendation, but I'm not saying that they are like absolutely not efficacious in that. There is some help in that, and there's a debate whether if it's better to have a probiotic in there, that you're consuming, or just leave the gut open for any other bacteria to come and colonize that. It could be a pathogen, right? What it's true, and you mentioned the prebiotic, it's important that my recommendation, until we have an effective way of actually degrading the antibiotics in the gut, so you don't lose the initial diversity, is that through your diet, during and especially after the antibiotic treatment, you can nurture those microbes back, and you can do it faster. So if you consume lots of plant fibers, fermented food products that would contain, important bacteria in there, that you have a diverse diet, that is free of highly processed foods, this would be a way of bouncing back from that loss of diversity. Of course, it's going to take some time, and it depends on the person. Some studies show that it can take a few weeks for some people, but for some others, it can take months or years before they reach the levels that they were before the antibiotic treatment. So that's why we want to prevent that loss in the first place.
Okay, so I actually do have a decent question for you. So how do you ensure that the resistance genes that you're introducing don't get remobilized to other organisms that happen to be in the gut, say by plasmid transduction or bacteriophage transduction?
Absolutely, that's a great question, and this is at the core of the safety of this technology. So what we have created are called engineered evolutionary safeguards, and our approach is that we have split the gene into fragments, right? And the result of this splitting is that it's harder to find the two pieces of the DNA, because eventually those two pieces can jump into the same bacterium, the unwanted bacterium, as you're mentioning, through a process called horizontal gene transfer. That is what you're referring to. The effect of splitting the gene into halves is more like, let's consider that the beta-lactamase is a shield, is a tool that you don't want other bacteria to use. So when you split them, if other bacteria pick it up, they don't know that this is a shield, right? So and they don't have the instructions on how to assemble it and how to use it. So through natural selection, this trait won't be advantageous for these bacteria because they don't have the time or the means of to make it useful. This is the concept behind it. Basically, these fragments, since bacteria cannot know exactly what they encode, they are basically invisible to them. This is our approach to prevent these horizontal gene transfer.
Thank you very much for an amazing talk. That's such a clever solution for this. My question to you is, when you're dealing with this, protecting the gut bacteria in the body, how effective is this going to be to protect it from, or going to slow down like resistance build-up? Is it complete, or is it only like a percentage? Like if somebody had to take biotics later on, would they be forever like immune to the negative effects?
That's a great question. So in here, what we're preventing is that we're going to generate new variants of genes that would target antibiotics that we currently use in the clinic, and that the frequency that we see those genes in the environment is going to be less. It's not going to actually prevent the emergence of resistance in a pathogenic group of bacteria that you're treating. If you think about it, there are like two mechanisms by which a bacterium can acquire a mechanism that confers resistance to the antibiotic. The first one is if you're a pathogen and you're exposed to the antibiotic, you can generate some mutations that are gonna make you not sensitive anymore to the antibiotic. The other way the bacteria acquire these mechanisms is what we were talking about with Ely, is that you can transfer genes horizontally from one bacterium to another, not from mother to sister, but from neighbors in the gut. That is what is called horizontal gene transfer. And in this case, it is possible, since commensals also encode these mechanisms of resistance. If you apply enough selective pressure to these populations, eventually you're going to select for one that is resistant to the antibiotics. And then, since microbes, the genes in the microbiome is basically a network, they can transfer these one to another, and they can land in a pathogenic bacteria. So with our intervention, we are suppressing this unnecessary selective pressure on the commensal bacteria of the gut. And in this way, we can reduce the rate at which we would observe the emergence of this new antimicrobial resistance. It is an indirect mechanism that is important to address.
Fascinating. Thank you very much.
Hi everyone. Hi Deep, Nissan, and then Dr. Shah and Serena. So we have 13 minutes left. So keep to one question, and so we can get to everyone. Thank you so much.
Thank you for this. I had a question related to the fact that, there's certain bacteria like H. pylori which are found in amongst a lot of people, and they usually do not give any problems, but sometimes they do to people. And so and then they are targeted, right? for people who have problems and they given antibiotics. But have you also found that maybe some of the bacteria that are targeted actually have certain other maybe factors which may not you know where you don't really want to destroy the bacteria completely because they have been sort of cohabiting in the human body for large populations over a very long time. So do they have any, maybe they have some preventive factors or things of that nature, but usually the treatment is tries to, I think, get rid of them. Any comments would be great.
Yeah, I think that this is an important point, and what you might be referring is to what we call pathobionts. So a pathobiont is a bacteria that normally would be inside you, and under certain conditions, it is not gonna cause any disease. But if those conditions change, then what what's supposed to be there in a safe way now becomes a bacteria that is problematic and that can give you, an infection and or some other health issue. For example, H. pylori can be like under some conditions, those populations can expand, and if you're susceptible in your genetic profile, it can lead up to cancer. So that's why it is important, that with this approach, specifically for gut pathobionts, we maintain the conditions as much as possible within the gut environment. So when you take the antibiotic, it's going to wreak havoc in there, and it's going to unbalance all the populations. And sometimes it can create the right environment, so these um, pathobiont populations can expand and be dangerous as well. Like that's the case, for example, of Enterococcus bacteria. Enterococci are usually present in the gut microbiota, but under certain conditions, they can expand and cause infections as well. So that's why with this approach, we want to uh, maintain these compositions and do not perturb these conditions that are very sensitive to those changes. That is a good point. Yes.
Thank you so
Uh,
much. Listen, yeah, go ahead.
hey, my question is, how do we know if a person's bacterial composition in their gut is good or not? Like, what are some ways that this is managed? Do we look at if the bacteria is producing nutrients or enzymes? Do we measure that from a sample or from their blood? Like, what are some ways that we can tell if the person has good gut bacteria?
That is a wonderful question, and you get to a really good point there. Um, we have not been able to come up with a definition of what is a healthy microbiome, what is a composition that is healthy for everyone. And the reason is because there's so much variability between different people because of their diets, because of their genetics, because of the environment that really, really changes the composition of the microbes that we have in there. There are some core functions that we know that are important for a healthy status. For example, the production of short chain fatty acids and the production of secondary bile salts are two, I think, important functions that now mechanistically we understand that must be present in there. But in general, the picture that emerges from all these studies that studying like different cohorts of, for example, healthy versus disease patients is that in healthy people, you see diverse group of microbes, like the diversity as a measure of how many different taxonomic groups you find in the gut is what really correlates to a healthy status. People that have less diverse gut microbiotas are usually correlated with disease status. The question there is always, what came first, the dysbiosis, this loss of diversity, or the disease, or is the disease inducing this dysbiosis? So it's a two-way road there that is still hard to define. But you touch upon there a good point, and in our approach that is also important because for most of people they have a composition that is defined and is specific for them, and for most of people it works fine. So we're trying to keep it that way, not changing any way. Just try to keep it this way by protecting them from antibiotics.
Okay, thank you.
Can I ask
So,
another quick question? Oh, sorry, please. Sorry, Dr. Shah, please
yeah, we
continue.
have Virgin.
Yeah, we have
Okay,
Dr. Shah, Serena, Gilbert, and Ripley, and we have around 5 minutes left, so please go ahead.
so first of all, thank you so much. And I had no time to, attending earlier. So my question from the, I mean, is about the facing or increasing the prevalence of the antibiotic resistance for the tuberculosis. And I was just wondering, do you have further information around that, especially about the immunotherapy? I mean, we know that some of the patients, they are struggling with this side, and I know that there is some researches recently developed around the tuberculosis and the effect of that in the immunotherapy for the patient who needed the immunotherapy. So I was just wondering, do you have further information to share with us or not? Thank you.
Yeah, thanks for the question. I think is the emergence of antimicrobial resistance, is directly related to the antimicrobial use. The more you use the antibiotic, the bigger the chances to increase the resistance. And this happens to infections of the skin, the lungs, as tuberculosis, as you were saying, as well as bugs in other sites of the body. You point out about immunotherapy, and I think if I have the connection right, is that one thing that we have seen that is very important, to respond to medical treatments is that your gut microbes, really control your immune phenotype, your systemic immune phenotype. So if you have the right group of microbes in your gut, and they are stimulating the epithelial cells and the mucosa in the right way, you can have better response to medical treatments. This is very well known for cancer, but now people are coming up with clever studies that really connect the gut with different systems of the body, with different organs, and one of those is the lung. So I would think that there might be a connection on how, if you have a healthy gut microbiota, maybe you're better at clearing out tuberculosis infections, or maybe creating a better environment to create a granuloma that would contain the infection more efficiently. So it is related to that, yes. I hope that answered your questions. I don't know.
Yes, thank you so much.
Yeah, thank you for this talk. I came late, so I'm going to yield my time. Maybe Gilbert or Grepley have questions. Thank you, though.
Thank you.
Do you have
Can
any
I just
questions,
quickly?
Gilbert, Ripley?
Okay,
Please
Gilbert,
go
yeah.
ahead if you do. No? Okay. Jamie, one question, and then Serena has one question, and then we'll close the room. Thank
Okay,
you.
I was just going to ask, and this is a total I don't know very much about this, but when you mentioned the lactotis, anyway, the bacteria you're working on, sorry, I forgot the word. Um, will this in any way affect people that are lactose intolerant, or will use a different bacteria, or is it any way connected to that at all?
In this case, it doesn't matter. the the name of the bacterium is Lactococcus lactis, but it doesn't have to be fed lactose to grow. So it is metabolically active in the gut, but it doesn't require lactose. So there wouldn't be any interactions with that for lactose intolerant people. That is not an
Okay,
issue.
thank you very much for indulging my question. Thank you.
Sure, no problem.
Yeah, Leslie, hi. You want to ask something?
Yeah, I'm sorry, I'm not quite sure this is in the form of a question, but I just really like the approach of protecting the microbiome that we have because I was just curious on your thoughts about the FDA regulation of uh, essentially microbiome regulating microbiome transfer as a equivalent in strictness to an organ transplant because on one sense it is good that we don't have a proliferation of poop milkshake sellers on every corner in advance of our knowledge. But on the other hand, it makes it more difficult to contemplate maybe shifting people's microbiomes because I work with movement disorders and Parkinson's disease, and
Hmm.
I have a mood disorder. So both of those chicken or egg, but show serious deficits in the microbiome. So um, yeah, so I really do appreciate just very very good approach to protect what we have if we can't have maybe the flexibility we want in shifting it at this point. Did you want to maybe my question is do you on the other side of the coin, being part of the field, do you know what efforts there are to make it easier to shift the microbiome as well as protect it?
Yeah, that's a great point, and I have a couple of minutes to answer it. Um, the 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 key here is that we are beginning to understand how to do that, right? And I think that will be the future, but we'll still like 5-10 years away from seeing a real therapeutics on that side. And the reason FDA regulates these microbiota transplants is because there are many unknowns on these. We have some known unknowns that we can control, but there is some other traits that can be transferred through the microbiota that we don't know of. So I think it's important to boil down to the particular members of the microbiota that can contribute to as you were saying, in the gut-brain axis that would bring responses to mood disorders or conditions related to the gut-brain axis, Parkinson's, Alzheimer. There are some recent studies that suggest that this could be the case. So I do think that in the future we could have some more defined approaches where we use not a single strain or just one particular type of bacterium, but either a combination of strains that we know that would work well, or as I like to see better, um, an engineered version of a strain that can, trigger the specific responses that would be therapeutic for different conditions. And I think we're going to see that in the next 5 to 10 years.
Yeah, thank you so much for answering all the questions. You had a hard stop at 5:45, so it's 5:46. So I think you have to go.
Yeah, thank you
Is
so
that
much, Katarina, and everyone here in the room. I really enjoyed these conversations, and you can always follow up with me. My email is in the slides that I provided, and here to the app of Clubhouse as well. Feel free to reach out anytime.
Yeah, thank you so much. We really appreciate that. And if you're open to it, maybe we can invite you back sometime, maybe in the late summer, fall or so.
Absolutely.
And
Yeah,
perfect. Thank you. It was
I'll
a
be
great
happy.
pleasure having you, and enjoy the rest of your day. And thank you so much. We
The
really
pleasure is
appreciate
mine.
the
Thank you so
time.
much.
Thank you, Dr. Andres. Take care.
All right. Bye
Thank
bye,
you,
Dave.
Andrew.
Bye,
Thanks,
Thank
everyone.
you.
Katarina, for the room.
Bye. Yeah, thank you
Thanks.
everyone for coming, asking questions. And as I said, as Andres also mentioned, you can follow up with questions directly or send them to me. And we can also set up another follow-up room, maybe in the fall when we have some availability again. And yeah, thanks. for coming. We have another room tomorrow at 9 a.m. EST, actually. It will be a really interesting room with Dr. Otti from the UK, and he will talk about how they created in the lab two quantum time crystals. We had Dr. Pedram here before, for, the Google senior scientist, talking about the time crystal, and they are basically continuing that research. They managed to have two time crystals at the same time and looking at the dynamics, but he will also talk more about how you can melt a time crystal and maybe have a time crystal liquid. Um, so it will be an amazing room, but it's at 9 a.m. EST. But we will record it for people and on the West Coast. It's really, really early. Yeah, it will be exciting. And then we also have another room tomorrow with Dr. Jill. No, on Friday. I'm really sorry. On Friday, oh, I'm totally confusing it. So that one will be tomorrow at 11 a.m. EST. So it's not as early. So 11 a.m. EST with the time crystal room. And then on Friday at 9 a.m. EST will be Dr. Jill. He will be talking about a multi-omic rejuvenation approach of human cells, which is a really interesting breakthrough in rejuvenation research. And then on Friday, we have another room at 9 p.m. EST, and Dr. Atta created basically a simulation of a time machine to study ancestor galaxy time life cycles. So basically to look back in time how galaxies looked and were right in the beginning of the time of the universe. So yeah, please, if you like discussions like these, feel free to come back, and thank you everyone for coming and asking questions, and hear you all soon again. Thank you everyone. I'll close the room in 3, 2, 1. Bye everyone.
Bye, everyone.